The spinal cord is probably the most important part of the human body, and it helps us perform trivial activities such as walking, standing, or even running. But what happens when damage occurs at this level? This is David, a young patient that suffered from paralysis for more than 7 years. Due to a spinal cord injury, the most important part of his body was interrupted, and therefore signals coming from his brain could not reach his lower limbs anymore. Seeing this picture, some might say that unfortunately he was sentenced to live the rest of his life in a wheelchair. However, with an incredible will from his side, months of treatment and intense recovery, David was able to make small steps again. His treatment basically consisted in applying electrical signals close to the injured area in order to promote local restoration of the neuronal circuitry and therefore send the required signals to his lower limbs. This technique is well known as the epidural spinal cord stimulation, and in many cases it has been proven to be very effective. However, because it's a non-specific type of stimulation, unwanted areas might also get activated, and the result, or the walking pattern, is not ideal. So if the ultimate goal is to provide a high life quality for patients like David, we need to do more. It is of great importance to create tailored treatment plans for each individual, and to do so, we need devices that can precisely target the nerves within the spinal cord, and in the same time record the stimulation efficacy, and thus avoid this unwanted stimulation and improve the walking pattern. Unfortunately, this is not possible with the currently available devices, and it is also not possible with electrical stimulation due to its non-specificity. So we have to look into other ways of activating the biological tissue. For instance, optogenetics, or in other words, the use of light for generating action potentials. This technique has been gaining a lot of interest in the field of neuroscience, and this is due to its great advantage of activating the tissue in a more type-specific manner. This means that only cells that are responsive to a specific wavelength will be activated and therefore action potentials generated. This is a very important aspect, and not only when it comes to delivering tailored treatment plans for patients like David, but also for research purposes, to get a better understanding of the mechanisms that occur within our nervous system. In order to quantify the effects of this new technique and to acquire relevant information, electrical recordings at the site of stimulation have to be performed. To do so, and to achieve both optical stimulation and electrical recordings, new electrode arrays have to be developed. Electrode arrays that are optically transparent and electrically conductive. And the most suitable material for that is graphene, a novel, highly conductive, mechanically stable, and transparent material. But apart from that, the comfort of patients like David is as important as the stimulation technique used. And in order to achieve this, all these bulky, big, and stiff devices have to be replaced with much smaller, biocompatible, and more flexible ones. To fulfill all these requirements, the envisioned structure of our system consists of transparent and conductive graphene electrodes encapsulated in a flexible, soft, transparent polymeric material, and with all electronic components embedded in small and thin chips. Using cutting-edge microfabrication and assembly technologies, we at TU Delft managed to fabricate on a large scale the first highly flexible graphene-based passive and active spinal cord implants that had a transparency rate of 80% and that showed no deterioration over time in biological environments and also no deterioration over time when bent. This was a great achievement for us, first of all from the research point of view, but it is also a great step towards providing a better life for David and patients like him that are maybe dreaming of walking again as they once used to do.